Assalamu alaikum girls. Today I'm going to start lecture on the characteristics of enzymes. As yesterday we stopped the lecture over there. I have taught you about what is the basic nature of enzymes. We also call them biocatalysts, and we have studied mechanism of action. And yesterday I told you about the structure of enzymes, which is like a globular protein. And I told you that they are made up of linear chains of amino acids like this. And then these linear chains of amino acids fold in a three-dimensional way to become like this. And it's a generalized picture to tell you about three-dimensional folding of enzymes, just like other proteins. Enzymes are also protein in nature. So let's move to the next characteristics of enzymes. And I also covered few of them, that all of them are made up of amino acids, and enzymes speeds up the reaction million times faster than uncatalyzed reactions. So what a good thing to use, million times faster. So enzymes are also not consumed during the reaction. And I told you substrate specificity of enzymes, that this bind to a very specific substrate like a lock and key. And now the next point today is only a small portion of enzyme molecule is directly involved in catalysis. Not every part of enzyme is involved. That small portion which is actually involved in the reaction or catalytic activity is called as active site. Active site is the one which has specific geometric shape, which has specific three-dimensional shape, which recognizes and binds substrate, specific substrate. If we look at this diagram, the blue color thing is enzyme, where the active site is shown with almost red line. This red area has a three-dimensional shape. And this is actually the region which is going to perform catalytic activity, which is going to change the structure of substrate or will speed up the reaction by converting the substrates into product. So you must remember that active site is actually involved in catalytic activity. And we will discuss later on that how active site can work. Okay. Now these are the next points of characteristics of enzymes. Enzyme production can be enhanced or can be lessened according to the demand of the cell. I mean, you can increase their production, you can lessen their production, and it is done by inhibitors and activators. Now the question is, what are inhibitors and what are activators? Inhibitors are the substances which slows down or prevents a chemical reaction. When there are different kind of chemicals which can either enhance the activity of enzyme, but here these chemicals are, which we are calling them inhibitor, they are slowing down or preventing the reaction totally. So, and there is another set of chemicals which we are calling them activators. As the name shows, these are the molecules which increase the activity of enzymes. I mean, there is not only one activator in nature, there are many. So these chemicals can increase the activity of reaction, but it depends on the demand. That if we want to enhance the production of enzymes, then the cell will use activators. But if the cell wants to lessen or totally stop the production of enzymes, then cell will use inhibitors, right? Next. Some enzymes do not need any additional component to work. I mean, they work on their own, but there are certain enzymes which require additional components, non-protein molecules or ions, which are called as cofactors. Cofactors have different nature. They can be inorganic or organic. You know what inorganic things are? The chemical compounds other than carbon are called as inorganic compounds. So among inorganic cofactors, metal ions are used. And among organic cofactors, the examples are flavin and heme. Some of the examples, right? And organic cofactors can further be classified as coenzymes and prosthetic group. If an organic cofactor is tightly bound to enzyme, then it is called prosthetic group. But if an organic cofactor is loosely attached, it's called as coenzyme. Now this is the easy way to remember this point. I am going to tell you the theory again as mentioned in book, but kindly note down this flow chart to easily learn this point of cofactors. As I told you, that if organic cofactors are tightly bound to enzyme, remember the cofactor is binding with enzyme, then it is called as prosthetic group. If the organic cofactors are loosely attached with enzyme, they are called coenzymes. The function of the coenzyme is to transport chemical group from one enzyme to another. Different kind of chemical group. You know, there are chemical groups like alkyl group or carbonyl, carbonic acid, or different kind of bonds. Yeah, radicals. So according to the demand, coenzymes transport chemical groups from one enzyme to another. Some important vitamins are also acting as coenzymes, like riboflavin, thiamine, and folic acid. They are the example of coenzymes. See, this is all about cofactors and coenzymes. The next point of characteristics of enzymes is that several enzymes can work together in a specific order to make a metabolic pathway. What is metabolism? I told you earlier. Series of chemical reactions. So look, here the series of chemical reactions are called as metabolic pathways. We can even name it, but here I have mentioned a totally journal reaction, a symbolic representation, so we cannot name it. But in nature, we can name those metabolic pathways. In a metabolic pathway, one enzyme takes the product of another enzyme as a substrate. After the reaction, the product is passed on to the next enzyme. Look at this generalized flow reaction. In fact, series of reactions. The first reaction is from A to C. A is a substrate and C is a product. The second reaction is C to E. Third reaction is E to L. Fourth reaction is L to M. And the letters Z, X, Y, and P are represented as enzymes, right? I just want to clarify this point, so that's why I made this pathway to tell you what this point meant. So according to this point, when a reaction A to C is occurring, Z is the enzyme. But when the second reaction starts, C is the product of first reaction acting as a substrate in the second reaction. In the second reaction, C to E, C is substrate and E is product. While in the third reaction, this E is now not acting as product, rather it is acting as substrate. So in the third reaction, E is substrate and converting into product L. Now L is substrate over here in the fourth reaction. So the product of one reaction is acting as a substrate in the another reaction. Assalamu alaikum Hamza. You're again awake. So hi. मेरा बच्चा अच्छा चलो. So I just want to tell you कि these are the series of reactions which are taking place and in which a product of one reaction is acting as a substrate in the next reaction, and then product of that reaction is acting as a substrate in the next reaction. So this is how metabolic pathways are created, and many enzymes are working together in specific order, right? That's all about this slide. Now we are going to proceed to uses of enzymes. Enzymes are used in food industry, brewing industry, paper industry, biological detergents. Obviously, we are talking about industries, man-made industries, but the enzyme being used in a human body and in animal body are playing tremendous role. We are not talking about those roles because that is going to be a whole book of knowledge. In fact, books, lots of books can cover that knowledge, but we cannot cover it in ninth class. So we are talking about industrial use of enzymes. In food industry, enzymes are used to convert starch into simple sugar. Starch is a polymer, a polysaccharide, converting into simple sugars, which can be used in making of white bread and buns. While in brewing industry, which is used to where we make alcohol, enzyme break starch and proteins into smaller compounds, smaller carbohydrates and amino acids. The products are used by yeast to produce alcohol. Yeast is undergoing fermentation to make alcohol, and yeast is a fungus, so it needs smaller molecules to digest. The enzymes are used here to break bigger molecules like starch and proteins into smaller ones, and those small molecules are used by yeast to produce alcohol. And this is happening in brewing industry. What is happening in paper industry? Here enzymes are breaking starch to lower its viscosity. Now the question is, what is viscosity? Viscosity is coming from the word viscous, means thickness. You know, I think I might have told you about glue, how thick it is. And let's talk about water. Water is running. Let's talk about juice. They are also runny. They are not thick. If you talk about soup, soup is a bit thick. It depends. Sometimes we make very less thick soup. Sometimes we make a very thick soup. That is what we call it as viscosity. So here in paper industry, enzymes are used to lessen the viscosity, which will help in making paper, right? Let's talk about biological detergents. Um, I'm sure biological detergents must be expensive because here we are using enzymes to remove protein stains from clothes, and certain enzymes are used in dishwashing to remove starch residues. Mostly in detergents, normal detergents, we use chemicals, but biological detergents must be environment friendly. So protease enzyme are used to remove protein stains from clothes because protease acts on proteins. And amylase are used in dishwashing to remove starch residues like carbohydrate residues. Okay, this is how enzymes are used in food industry, brewing industry, paper industry, and in biological detergents. So this is all about uses of enzymes. Today I covered the lecture quite early. I thought it will take too much time to cover these characteristics of enzymes and uses of enzymes. So I am ending my lecture here because I wasn't expecting this quick discussion of characteristic of enzymes. So today, this is all about today's lecture, and Allah Hafiz.